Hi everyone, my name is Eirini Tsirvouli and today I'm going to present your work on the logical and experimental modeling of psoriatic keratinocytes. Psoriasis is a chronic skin inflammatory disease which is characterized by continuous interplay between immune cells and keratinocytes. During the later or chronic stage of the disease, T helper cell populations are dictating the cytokine microenvironment in psoriatic lesions. Namely Th1 cells, which mainly produce interferon gamma and TNF-alpha, and Th17 cells, which mainly produce interleukin-17 and 22. Keratinocytes, in response to these cytokines, appear to be hyperproliferative, resist apoptosis, and undergo aberrant differentiation. Additionally, keratinocytes secrete various ligands, mainly cytokines and chemokines, that recruit even further immune cells and promote their survival and proliferation. Of special interest in this project was the potential regulation of CPL2 alpha in response to these cytokines. CPL2 alpha is a phospholipase which produces arachidonic acid, a precursor of lipid pro-inflammatory mediators called eicosanoids. For that reason, my colleague in the lab characterized the regulation of main eicosanoids by IL-1722 and found that PGE2, or prostaglandin E2, was upregulated in response to these cytokines. As every other eicosanoid, PGE2 undergoes a complex metabolism and that we fully described by their gene expression. And we also showed that EP4, one receptor of PGE2, and the only one which is actually directly connected with hyperproliferative signaling pathways such as PI3K ACT, was upregulated in psoriatic keratinocytes. Inspired by this newly described signaling and also by prior knowledge, we present you the psoriatic keratinocyte model. The model has as input Th17-derived cytokines, IL-17 and 22, Th1 cytokines, TNF-alpha and interferon gamma, and also EP receptors. The model is able to represent the main physiological states and phenotypes of psoriatic keratinocytes. And it does so by looking at the behavior of phenotypic markers, either regarding keratinocyte cell fate or markers that represent ligands that would be secreted from keratinocytes in response to certain stimuli, then they would act on either keratinocytes themselves or other cell types and mainly immune cells. In order to validate the model, we we measured the expression of 17 of these phenotypic nodes, spanning across all sets of phenotypes and physiological states. The conditions that we looked at the state of these nodes was in 3D Hackett cultures stimulated by IL-17 and 22, and 3 chemical perturbed conditions where we had CPL2 alpha inhibition, calcipotriol, which is a frequently used and effective anti-psoriatic, and their combination. The model appeared to have very high accuracy with 59 out of 68 states of these nodes being correctly predicted. Additionally, in the lab, we we look at the physiological states of the 3D cultures, namely their proliferation and differentiation, and we failed to see this hyperproliferative behavior of keratinocytes as has been previously described. The truth is that these cell models that are currently used in psoriasis are not adequately representing this nature of keratinocytes, are rather more a differentiation model and more complex and hard to recreate in the lab cell models are used to adequately describe this phenotype. On the other hand, the computational model adequately described this hyperproliferative response of keratinocytes to these cytokines as it was previously described in clinical studies and in the literature. Having the confidence of having a well-predictive model, we wanted to explore this CPL2 alpha PG2 EP4 axis in conditions that we did not check in the lab. For that reason, in addition to the TH17 cytokines that we did for the validation, we looked at the behavior of cytokine of the keratinocytes in response to TH1 cytokines plus EP4, and the condition where all cytokines were present and EP4 was active. The same chemical perturbation were also simulated. This is a heat map of the stable states of the model. Here we see that the first three rows represent cytokine stimulated non-chemically perturbed conditions. AVX stands for Avexin 001, which is the drug we used for CPL2 alpha inhibition, Cal for calcipotriol, and combo for their combination. The stable state analysis revealed that the two drugs appear to have two different modes of action, where Avexin is actually affecting mostly proliferation markers, while calcipotriol on the other side appears to restore more the differentiation phenotype and also promote apoptosis or sensitize keratinocytes to this process. The most interesting thing was that the combination of the two drugs, even though they had two different modes of actions, appears to be the only case where we see a completely shutdown of proliferation markers. And also we see the inhibition of main cytokines for psoriasis, such as IL-6 and TNF-alpha, both described to synergistically promote together with other cytokines, the psoriatic phenotypes. As a next step, we wanted to explore a bit more the influence of each set of cytokines into the system. And we did that so with value propagation, which looks at which nodes are bound to be active or inactive in response to certain stimuli. There, we show that TH17 derived cytokines actually uniquely control the state of many differentiation and proliferation markers, as we have seen before in the literature, and TH1 on the other side appear to have a more stronger control and unique control over immunomodulatory and regulatory nodes. At the same time, it was the majority of the nodes was controlled by both sets of cytokines, something that supports the hypothesis that psoriasis can not only be attributed to single cytokines, but it's their synergy that actually ends up to the disease. Further, using the model as a hypothesis testing tool, we wanted to check why interferon gamma and TNF-alpha do not promote apoptosis in keratinocytes while they have been described to do so in other conditions. Here we did some stochastic simulations with the probability of all nodes being equal to be active or inactive. The results were multiple possible stable states, but they could very easily be distinguished to states that would be proliferative or survival states and apoptotic. An analysis of these states showed that the expression, the activation of SOX1 and SOX3 was related to more survival states. Interestingly, it was also found that SOX1 and SOX3 are overexpressed in psoriatic keratinocytes. When the model was being built, it was found that SOX1 and SOX3 are actually involved in a negative regulatory loop with their activators STAT1 and STAT3. However, this negative feed-forward loop was found to be impaired in specifically psoriasis. This gives some information that it is possible that psoriasis is not only being caused by many positive feed-forward loops. However, we see an importance of some impaired negative feed-forward loops that would otherwise shut down the inflammatory response. As a last part, we wanted to use the model to predict those perturbations, activating or inhibiting a single or combinatorial that would help restore a normal state of our markers. The perturbations consisted mostly of known targets or very important nodes in psoriasis, and that's of course a good sign that the role of these specific entities is adequately described in the model, but also proposes EP4, a target, and peer protein kinase C, a protein kinase alpha, as a possible target of psoriasis that should be further explored. Wrapping up, in this work, we show how CPLA2 alpha, PGE2, EP4 axis might be involved in keratinocyte role in psoriasis and how its inhibition alone or in combination could be an effective therapy against the disease. We show that TITS1 cytokines have a more immunomodulatory role, while TITS1 derived cytokines are controlling mostly keratinocyte cell fate. We showcase how logical models can give mechanistic insights in stimuli and perturbation effects. We show the importance of contextualized interactions, such as the case of SOX1 and SOX3. And we propose the role of EP4 in psoriasis that should be further explored. Thank you for your attention.